go ahead. It's great to have this opportunity to share my work. And today my talk is Deep Sea in situ term called omics, review microbial dynamics. I'm Dr. Wang from the Institute of Deep Sea Science and Engineering, Chinese Academy of Sciences. So I'm now in Sanya, Hainan Island. Now here is the outline. First, I will introduce two questions and one bottleneck problem for deep sea microbial studies. Then second is facility development for deep sea in situ work. Then the third one is a time course in situ study. So this is some preliminary result from my group, and it's not yet finished. First question is about, is there a microbial rhythm in Twilight water column? We know that there are clear rhythm for microbial work, particularly for cyanobacteria expression. This is published in Science 2014. And in the sea surface, there are clear rhythm for the microbial expression and other activity. But how about for the deeper ocean, for example, 500 meters and 1,000 meters? Clearly, it seems that the current studies showed no such rhythm in the deeper waters. So there are all, for eukaryotes in the surface water, there are also some rhythm. It's very clear. But for deeper waters, we don't know. But we know there is sufficient production. About 20% of the sufficient products can reach to 1,000 meters. We know there are a lot of sea snows. So they are input into the deep sea to support the heterotrophics, so that they can also respire these organic products into carbon dioxide and to release some nutrients to the deep ecosystem. So the autotrophs such as ammonia oxidizing archaea and nitrate oxidizing bacteria can be the major autotrophs in the deep waters. So enter the viral shock and also other breathers, for example, nanoplanktons. So they can use the label carbons produced by the autotrophs to support the deep ecosystems. So there are some internal carbon cycles. So in the deep waters, so we know that, but we don't know whether the microbial communities and activities can be affected by the organic input. So this is an interesting hypothesis. The next question is how to quantify these microbial processes involved in the element cycling, particularly carbons. We know that the greenhouse gases can be absorbed in the surface water, and the water can flow into the deep waters with the sea snows and some megafaunas, the migrations to deep water. And now we know that in the global oceans, we sampled in many sites, so we know there are communities. But how to quantify these microbial processes? For example, how many tons of carbon dioxide and methane are biologically assimilated? So these are very important questions for under this global warming background. So, and there are a lot of several works, they have done a lot of things to answer these questions. For example, the Global Institute, they published a work in Science. So they show the nitrate oxidizing bacteria contribute a lot to deep sea organic carbons, up to 40% of organic carbons in deep water. So later, they use isotopic cultivation in deep waters. So this is their method. Then later, Dr. Zhang from Xiamen University, they published work in PNAS. So they also showed a lot of equations to quantify this nitrate oxidation rate and ammonia oxidation rate. So their work clearly showed that nitrate oxidation rate outcompete ammonia oxidizing process in the organic production. So only this is not clear, obviously not enough. We can also use omics to detect what are those autotrophic microbes are working in the deep water. So what are those carbon fixation process in the carried by these microbes? So this work is published in Cell. They use Tara Ocean samples, so they generate a lot of genomic data and use the transcriptome to quantify these processes. So we know what are those genes for carbon dioxide in deep water. So what are those microbes? So this is greatly expand the spectrum of microbes and the process for deep sea carbon turnover. Now, this work only expand the space axis, but for time axis, we want to know how under the continuous time course, how these microbes can react response to these environmental changes and organic input from surface. So the future direction is that we can study both the multiple spatial and the multiple time course activity of these microbes in the deep ocean. So we also need some long-term in situ platform to detect the variations of the genes, microbes, their correlation and interactions. So we can also quantify this the deep water ecosystems, particularly for this megapaunas, microplanktons. So we can modeling and for the nutrient flow. So bottleneck problem, we know that for deep water microbial work, the challenging work is that when we take sample from the deep water to our research vessel, the environmental changes can affect our sample. For example, the temperature, pressure, and oxygen can also change greatly, and we cannot avoid some contaminations when we're handling these samples. For example, the community will change. For example, in deep waters, if we cannot kill these microbes, so they can grow, the community changed, although we can extract DNA from the samples. The next is transcriptome change, under different stimulus that the environment under changing environment. So the microbes express a different profiles of microRNAs. So next for the other deep sea megafaunas, their morphological changes we cannot recognize there for taxonomic work. The next for fish, is there physiological conditions also? It's not the same as them in the deep water. So we need some in situ experiment for deep sea life sciences. So here is an example that we take pushcars from deep sea. So when the pushcars return to the sea surface, we can a lot of bubbles released from these pushcars. So if consider that if we do the bacterial cells, there are a lot of dissolved water gases in the cells. So they will explode when they return to the surface. So we cannot see a lot of microbes. So you just cannot catch them when you filter the sample. So later, the second part is facility development for the deep sea in situ work. So now there are a lot of large volume filtration and fixation apparatus, and there are also a lot of multiple filtration apparatus. So they're developed by the WHOIS and other institutes. Then below there are ISMIF and MISNEC. They're developed by my institute. They can also fix and filter the microbial samples in situ. And as far as I know that this platform, ESP and the DESP, in details, it's an environmental sample processor. As the first in situ platform for deep sea research, we can filter the samples and lysis the cells, and for DNA extractions, later they can use a qPCR and a gene array to detect the gene expression then in time course. Then let me first introduce this ESMIF in my developed by my group. So we can in situ filter the seawater first, then use preservatives to fix and kill these bad microbes in the filtration chambers. So my work using tidal waters can demonstrate that there are a lot of differences in the when we use Niskin bottle and ESMIF. So in the Niskin bottles, we can find a lot of cyanobacteria. So this means the bottleneck perhaps have opened when they return to the ocean surface. So a lot of cyanobacteria were introduced to the sample. So next one is a project led by myself under the support of National K Research and Development Program. It's a Phoenix in situ platform. So it can be equipped with many scientific facilities. So first, for example, there are laser 3D imaging for ecological observations. There are also microscopists and microbial fluorescence counters. For molecular work, it can be equipped with the nucleic acid collectors and gene sensors and Raman spectrometers. So there are a lot of, it can load about 200 kilogram facilities. So the working depth is about 3,000 meters. So it can be carried by the submersible between different sites, can move around the cold seas to collect samples at different time course. Now here is how the MISNEC works. It's a multiple. There are a lot of there are about 9 working units, so it can collect samples from at least 9 time courses samples in situ. So here is a host connectors. When one sample is collected, it can shift to next one. So connect with the host connector. So there is a switch box, can switch the host to different filtration chambers. So at the end of this apparatus, there is a nucleic acid column collectors. So after cell lysis, and we can collect the nucleic acid directly at the end. So first, it can filter seawater into the filtration chamber, and the lysis buffer where it can be injected into the filtration chamber. Then later we mix this lysis with alcohol, so nucleic acids can be absorbed into the silicon bins. So later on board, we can wash off this nucleic acid off. So for the non-polar sequencing, Illumina sequencing, here is how this MISNEC mounted on Phoenix lander. And the working unit can be easily rotating in the deep sea set. So after one working unit is finished, so the next one move into the host connectors. So we collect samples for the next time period. The next part is is a time course work carried out by my group. First, the experimental site is offshore of Hainan Island. The depth is about 1,000 meters. So we did 4 deployments of lander. So each for this winter. Another one is for me summer in 2020 June. Okay. Okay, we measured the environments. For example, the oxygen. We can see in the first deployment, the oxygen concentrations fluctuated. Here you can see about 4-fold increase of oxygen. For the next day, the oxygen concentration did not change. For methane concentration, you can see for 1,000 meters, there are some irregular changes of methane flux. So, but there is no, we don't know how, no reason, for the methane flux. We also record the turbidity, sedum, and chloride in another cruise. So you can see the turbidity, sedum, there are no regular changes in the deep water. See the turbidity. Here is landing. When the lander landed on the seafloor, about 3 minutes, there was a high turbidity caused by the landing. But later, we cannot see any turbidity in the seafloor, so in the water. Now let me introduce the sampling information. So for the MISNAC, for each collection unit, we filtered 80 liter bottom water. So it's a DNA RNA co-collections. So we made 2 dives for continuous time period. For this, we also take Niskin and eSmith as control. So using Niskin, we took 10 liter winter samples for filtration. Here are the time periods. For winter sample, we sampled in 6 time period. For the winter sample, for the summer samples, we took samples from 3 time period. After the MISNAC work, so we washed off the nucleic acid, and then we separated DNA and RNA by enzyme digestions. For the DNA, we use Illuminer and nanopore for metagenomes, and for the RNA, we made metagenome, metatranscriptome data. So after quantification and the decontamination, we have 22 metagenomes and 22 metatranscriptome and nanopore data. Larger one TB data. It's a huge data set. So next, let's have a look at the prokaryotic communities. It's based on the 16S gene race we call MITAC. So we compare the microbial communities between winters and summers, between metagenome and the metatranscriptome. The first is clear that if we use Niskin and eSmith unfixed samples, we can see a prevalence of autonomous in the sample, particularly in the transcriptome. And for the eSmith fixed, in this sample we killed the microbes in the sample with buffer. And for these samples, we can see all right, let's go to the PCA. Let's go to the PCA result. So we use the PCoA analysis to characterize their differences in community structure. For the 2 metagenomes, we can see the summer and the winter metagenomes. The microbial communities are quite different. Well, interestingly, their transcriptomes are quite consistent in community structures. And for the ismif and the Niskin samples, they stay away from the three group of microbials. So my primary conclusion is there are some dead microbes from upper layers. So although there are some differences in metagenomes, but their transcriptome are same in terms of microbial composition. Now we compare the biodiversities between winter sample and the summer sample. You can see both for Chao-1 and Shannon, the biodiversity winter are always significantly higher than summer samples. So this is perhaps because the nutrient poor conditions in winter cause a lot of different microbes using a wider spectrum of organics. We also detect the novelties of species in transcriptome. So the normal 16 MATEC rate is about 10. So this is about twofold higher than the novelties reported by Tara Ocean project. So this is perhaps because we sampled more waters using MISNAC, but perhaps not because Tara Ocean can also collected a lot of samples from surface water. So next is eukaryotic communities. First, we report there are a lot of ciliates can be collected by MISNAC, but for other method, if we use the in situ filtration, it's the ISBIF method. If we use the in situ cell lysis, there are a lot of, so this can also like in the MISNAC filtration chambers. So we use this sample also for the human sequencing. We detect the 16S MATEX only in the in situ DNA RNA collections. We found a lot of ciliates species. So this is very interesting. So what are they? Now we use the phylogeny to demonstrate that the dominant ciliate species is Oligotrichia. We cannot detect them with photos, cameras. Here are some images from their shallow water relatives. So in this tree, we can see that there is a considerable distance between our 16S MATEX with their relatives. So they are perhaps dark matter macroplanktons at 1,000 dips. So previously, we never detect so many ciliates in the deep water. So with our sample, we can also detect a lot of ciliates that were not so predominant in the Niskin and the ISMIF samples. So let's please note these two parts. First, we compared their compositions between metagenomes and the metatranscriptome. We found that for the ciliates, there are not quite many in the metagenomes, but there are a lot of their transcriptomes, a lot of their genomic reads in the transcriptome. For example, the intramacro nucleata is a diverse group that includes many common predators, marine ciliates. So this means for another group, they are polycystic, the group of radial lineages. So they are fossil microbes. They drop to the seafloor. We collected, but they are dead. So we cannot detect their RNA or gene expressions. So this is a PCoA analysis of the eukaryotic communities. So it's really different from the prokaryotic communities. We can see their winter metagenomes and the summer metagenomes. The communities are not far from each other, but for their transcriptome, we can see even for the winter samples, the internal differences are quite big. So in different time period, their activities differ greatly. Well, the next question is there a rhythm for deep sea microbial eukaryotes? You can see for the 18S OTUs from the MetaX, you can see always at 3 a.m. to 7 a.m. There is a prevalence of these two OTUs in our samples. So we don't know what makes their prevalence in the deep water at this time period. So in the 2 days, it's quite consistent. Now it's a functional annotation of the situ omics works. So we need first normalize the metagenome data using the single copy conserved genes. So the method is published in Nature Communication. It's a MOTUS2. So we normalize this data so we can quantify the functional genes in different time course samples. We need to also normalize the transcriptome data. Now we use 40 single copy conserved genes. So you see for the winter samples, the single copy conserved genes expression is much lower than the winter samples. We need quantification. Normalizations. So this is a PCoA analysis without normalization. So after normalization, we can see the samples from the same time period from different days, sampling days. So they stay together very closely. Also for the winter samples, except for three time period, there are quite large differences. So for the top 20 most abundant MAGs, so we detected their transcriptome levels after normalization. For the top one, most abundant species is anaerobic. It's a anaerolineness. So it's the most actively transcript in winter, in summer. And number 2 most abundant is Salasatrichium. It's an MG2 group bacteria. So it's very actively transcript in winter. So here we can compare their expression profiles in between winter and the summer. For this bacteria is really abundantly transcribed in deep sea in our sample. So next, we also find AOA, star 202 bacillus and Methylococcus are also comparatively higher in transcriptome of winter samples. Well, for the most active genes, we detect box B and the GNL genes for aromatic degradations, very active in the group B bacteria. And for this anaerolineness, they expressed ADHP for alkane degradations. So you can see for summer samples, this process is more active than the corresponding winter samples. Well, so next, let's have a look at the star 202 lineages. Well, this is a very important DOM degraders in deep water. So there are a lot of lineages in 2004, star 2004. So we can see for the 2 dominant lineages, 2 and 3, their expression profiles are quite different. So this means their ecological roles are also different. For the osmolyte metabolism, so mostly carried out by the lineage 3. And also for the DOM degradations, so lineage 3 are also the dominant players that degrading the DOM. So for the polysaccharide degradation, we reveal that the propionate bacteria can degrade a lot of polysaccharides, for example, chitin, cellulose. So with this method, we can see both in winter and summer, these bacteria can also is a dominant species that carry out this process. So next, we use the transcriptome to detect the C1 metabolic patterns. For both winter and summers, we can see the AOA carbon fixation by the ammonia oxidizing archaea. So in winter, the carbon fixation activity is much higher than the summer samples. And also in the early morning, from 3 a.m. to the 3 p.m. periods. So their activities are much higher than the other time period. So the difference is quite significant. So we also detect the methane oxidations, the same as the AOA carbon fixation. So they are very strong in winter and the morning time. And for the carbon monoxide oxidation, so this is also the case. It's very interesting. Well, now let's have a look at the correlation network between the different time period. So we correlate the gene transcript. So we can see the tool formed several modules. The most important one is module 8. It's a core of the network. And this one, you can see this one, module 8, is another important network. So it harbors a lot of genes for RNA synthesis, utilization of ammonia and proteins. So, and between the 2 days, you can see the genes within the modules are quite consistent at the different time periods in the 2 days. And we take almost all the important modules in the network. You can see in the 3 time periods from 3 a.m. to 3 p.m. in 2 days, they are quite consistent for almost all the modules. But for the next 3 time periods, the gene expression profiles differ much. So they are perhaps affected by the organic input from the surface water. So this input is perhaps affected by for example, the water current or sunlight density from the surface. Here is a network of species. In this network, the most important species are from Proteobacteria, Chloroflexata, and SAR 224. So they are very important players in the network. So they degrade or generate organics for other species in the network. So this is a brief summary of different roles of major microbes in the bottom border. First, there are a lot of DOM degradation bacteria, for example, SAR 202, anaerobic lineages, and this group, marine group 2 bacteria. And for the nitrogen cycling, they degrade a lot of glycine and serines to get carbon sources and also ammonias. So these ammonias are used internally by this bacteria for ammonia assimilation. So this ammonia can also be the source of AOA. So they use AMO genes to oxidize ammonia to nitrate. So nitrate can be also further oxidized to nitrate. So for the carbon cycling, we know that both AOA and AOB can produce labile organic carbons using autotrophic process. There are also perhaps an underestimated source of organics. They are using methanes as a carbon source. to generate organic carbons. So perhaps they are really abundant in the bottom water because they are very close to sediment. Our inlet water is just about 1 meter above the seafloor. So there are perhaps a lot of methane released from sediment. Well, using CBB cyclings, SAR 324 can also fix carbon dioxide to produce the organic carbons. Perhaps there are a lot of other carbon fixation process. We don't know, we are not sure. They are perhaps bidirectionally. They can degrade organic, can also fix carbons. We don't know. We have to use other isotopic labeling to detect the other carbon fixation process. So at the bottom, maybe there are very chance to degrade this, to infect these autotrophics or heterotrophics to release their organic carbons in the cell. So to support the deep sea ecosystem. Now here is the summary. First, there are perhaps a lot of ciliates. They are perhaps eukaryotic dark matters in deep sea zones. So before we have not got their images or genomes for this important group of macro planktons. So autotrophs are more important in winter than in summer. So this is because the organic input from surface is much less than in summer. So autotrophs are playing important roles in organic producing to support the single ecosystem. So this is maybe also the case in the morning time when the surface input, say, for example, marine snows are quite less than in the afternoon time periods. So there is probably a half-day shift of microbial communities. So depending on the surface water productions to for the deep sea. So the last one is the ecological importance of bottom water probably has been underestimated. For example, the methane assimilation could be important source of organics for the deep sea ecosystem. So acknowledgement. First, I would like to thank my group. They are very diligent and smart. For the next one is our engineer team. So they collaborate with me for 7 years. So next, some funding sources: National Research Foundation, Chinese Academy of Sciences, and the National K Research Development of China. Well, thank you. Thank you for your attention. Any questions?
Thanks. That was a really nice talk. I think we have